A warm welcome to you all to watch the process of fertilization in angiosperms. In the previous class, we discussed about the process of pollination, which is followed by fertilization in plants. Let us see what is fertilization or define fertilization. The fusion of male and female gametes is the process called fertilization. So, today's topic is fertilization in angiosperms. So, the name of the fertilization in angiosperm is double fertilization. Double fertilization. Define fertilization: fusion of male and female gametes to form zygote. Let us see how this process is taking place. The events of fertilization. Events. What are the events observed in the process of fertilization in angiosperms? The pollen grains these are the pollen grains. The pollen grains are deposited on the surface of the stigma. This is the stigmatic region, stigma. This is style and this is ovary. Here ovule, inside ovule, and this is the embryo sac. Now, the pollen grains, the first event is deposition of pollen grain on the stigmatic region. The next event is the stigmatic region, the pollen grains are started to germinate. So, first there is a deposition. If the pollen grains are compatible, it starts to germinate in the stigmatic region. Due to the presence of stigmatic fluid present in the wet stigma, there are two different types of stigma, wet stigma and dry stigma. In the wet stigma, it secretes a fluid. Due to this, the pollen grain starts to germinate. Next event is this germination. As soon as the pollen grain germinates, it starts to elongate. It grows along the length of the stylar region. Then there is a direction towards entry of the pollen tube through the micropylar region. This region is micropyle. Direction of pollen tube towards the micropylar region. So, these all events. Then the next event is when the pollen tube directed towards the micropylar region, it takes two male gametes. These are the two male gametes. The male gametes are enter into the embryo sac and it is discharge of this male gametes into the synergy. These all events we are going to learn in detail. First of all, there is the deposition of, the first event is deposition of pollen grain on the stigmatic surface. There are number of pollen grains, large number of pollen grains are dusted or deposited on the stigmatic region, but all the pollen grains are not able to germinate. When the pollen grain is compatible to the stigmatic fluid, it starts to germinate. So, first event is germination of pollen grain on the stigmatic region. So, it develops at the time of germination, it develops a pollen tube. This is the pollen tube. For the germination of pollen tube, there is a fluid substance present on the surface of stigma in the wet stigma, and there is a structure called pellicle which helps in the dry stigma. So, due to the presence of this, the compatible pollen grain which begins to germinate. So, there is a chance of compatible and incompatible. So, this stigmatic fluid or the pellicle which decide the compatibility and the incompatibility of the pollen grains to promote germination or to inhibit germination. Then, as soon as the pollen tube germinate, this pollen tube starts to grow along the length of the style. There are different types of style. Based on this, there are 3 different types of style, namely hollow style. The first type of style is called as hollow style, or it is named as open style, which is observed in monocots. The monocots which possess hollow style. This is the style or region. In the style or region, there is a hollow canal. This canal is filled with this, there is a hole in the hollow canal, and through this it is lining with a single layer of glandular tissue named as transmitting tissue. Transmitting tissue is present in the hollow canal in the style or region. The type of style is called as open style, or it is called as hollow style. So, it is lined with glandular tissue which is named as transmitting tissue. The transmitting tissue is filled, it helps the pollen tube to germinate. This it gives the nutrients because it's filled with this lipids, carbohydrates and some kinds of enzymes. So, it nourishes this developing pollen tube. Then another type of style which is observed in dicots. In dicots, the type of style is named as solid type, or it is named as closed type. Closed type or solid type style. Here, this entire style which is filled with the transmitting tissue. A core of transmitting tissue present on the style or region observed in solid style in the dicots, that is solid style. So, the function of the solid style is, the transmitting tissue is similar to the function of this hollow style, the transmitting tissue. It nourishes the developing pollen tube, because the pollen tube in this solid style, the pollen tube grows along with the intercellular spaces of the central core of transmitting tissue. The difference among these two is, the hollow style, there is a single layer of glandular tissue called transmitting tissue, but solid style, there is a central core of transmitting tissue. So, when the pollen tube germinates, it grows. When it grows along the style, it grows along the intercellular spaces of this transmitting tissue. Another type of style is named as semi-solid type or half-closed style. So, this is intermediate between the solid type and the hollow type, the open type and the closed type. In between, this is the intermediate between the solid as well as the hollow style. So many authors explained and gives their opinion regarding this transmitting tissue. The transmitting tissue only present in the solid style alone, but certain authors, their opinion is it's present in the hollow style also. So, presence of transmitting tissue in hollow style as well as solid style, there are different opinions among the scientists. So, this is the intermediate between this two, hollow and solid type, is called semi-solid type of style. Now, the next event is, first event is deposition of pollen grain on the stigmatic surface. When it is compatible, the pollen grain germinates. Second event, growth of the pollen tube. The pollen tube grows. Once the pollen tube starts to germinate, there is a elongation of pollen tube. It grows along the length of the style and the ovary, and it enters towards the micropylar region. So, next process is direction towards the micropylar end. The pollen tube, entry of pollen tube. Now, the entry of pollen tube directs or guides towards the micropylar region. through this there is a structure called obturator. So, with the help of this obturator, it leads the pollen tube towards the micropylar end. So, when the pollen tube germinates, at first here when you look at the structure of the pollen tube, here there is the tip region, the extreme tip of this region, which is hemispherical in structure. So, when the pollen tube germinates, the pollen grain, the pollen wall, outer wall, there is a pollen wall. This pollen wall discharge all its content to the pollen tube, and this cytoplasmic content which is deposited in the extreme tip. So, it is called as callose plug. Callose plug, the cytoplasmic content which deposited in the extreme tip of the pollen tube, and the remaining part of the pollen tube there is a vacuole. So, this vacuole is cut off from this callose plug, and now this part is the tip part is called as cap block, tip of the pollen tube. So, this is the tip and this is the pollen grain, tip of the pollen grain is cap block, and the pollen tube elongates. When it grows, it brings two male gamete, these are the two male gamete, and a vegetative nucleus, vegetative tube nucleus, and all the contents are carrying along with this pollen tube. So, now direction towards this micropylar region, this region is the micropyle. Due to the guidance of obturator, this pollen tube, it grows along the length of the style and direct towards the micropylar end. Now, from this discharge of pollen male gametes, discharge of this content into the embryo sac. This is the region embryo sac. Embryo sac, which consists of 3 antipodals and 3 synergids. These are the antipodals and these 3 are the synergids. Now, the pollen tube carrying 2 male gametes. These 2 male gametes are discharged into any one of the synergid. That is, normally it is entered into the egg. The center there is a egg. This one of this male gamete which fuses to the egg. So, entry of pollen tube. Before that, the entry of pollen tube also there are different types. So, now direction towards the micropylar region through obturator. When this is carrying, when the male gametes are carrying towards this embryo sac, the entry of pollen tube. Next process is called as entry of pollen tube. Entry of pollen tube. The pollen tube either enters through the micropylar region. So, it is micropylar entry. It enters, either it enters through the micropylar region or it enters through the chalasal region. This region enters through the chalasa. So, look at the diagram here. This is the embryo ovule which contains embryo sac, in the embryo sac, egg, and these are the synergids. Here is the antipodals and here is the polar nuclei. And now here entry of pollen tube either through this micropylar region. If it is entered through the pollen tube enters through micropylar region, or it enters through the chalasal region, or it enters through the integuments. Either it enters to micropylar region, or it enters through chalazial region and reaches the egg, or it enters through the integuments. These are the integuments. Through these three different entries are possible. Now we are looking this diagram as entry of pollen tube towards the micropylar region. So now it carries two different male gametes. Now this process of fertilization is double fertilization, because fusion is two times. Fusion of fertilization, fusion of male gametes. It carries two different male gametes. One of the male gamete which enters and fused with this egg. Fusion of one male gamete with this egg is, and it formed as zygote. So this process is known as syngamy. The name of this process is syngamy. What is syngamy? Fusion of male gamete with the egg. Now another male gamete is there. This male gamete fuses with the polar nuclei. In the center of this embryo sac, this is the embryo sac. Embryo sac which consists of three different regions. The antipodal region which contains antipodals, and this region is synergid. In the middle there is an XII, and here there is a polar nuclei. Sometimes the two polar nuclei already fuse together and forms as a secondary nucleus. So this male gamete, one fuses with the egg, that is called syngamy. Another one fuses with this polar nuclei or the secondary nuclei. So there is two times fertilization taking place. Two male gametes which fuses twice and forms two fertilization. So this process is named as double fertilization. It is the important event observed in angiosperms. Double fertilization. Two times fertilization taking place. Let us see in detail about this double fertilization process. Double fertilization which is observed by S.G. Navastin and L. Gainard in the year 1898 and 1899 in Lilium and Fritillaria. In Lilium and Fritillaria, S.G. Navastin and L. Gainard in the year 1898 and 1899 observed in Lilium and Fritillaria. There, here is the diagram of pollen grain. This is pollen grain, structure of pollen grain, germinated pollen grain, and this is the structure of ovule. The ovule consists of embryo sac. Here the pollen grain, when the pollen grain germinates, it produces two male nuclei and a vegetative tube nucleus. The male gamete is N. The male gamete or male nuclei is N in number, haploid. Likewise, the vegetative tube nucleus also haploid in nature. And in this embryo sac, this is the embryo sac which consists of antipodals, egg apparatus, that is egg apparatus which consists of synergids and the egg, and the polar nuclei. This polar nuclei, either it is polar nuclei or already it is two polar nuclei fused together and forms as a secondary nucleus. So one fusion already taking place in the polar nuclei, two polar nuclei fused together and forms as a secondary nucleus. Now from here there are two gametes, male gametes, that is male nuclei, haploid. One of this male gamete fuses with the egg of this synergid. Here from this egg, this male gamete fuses. So N plus N, this XL also N, male gamete also N. So N plus N, it forms as a zygote as 2N. This process is known as syngamy. Syngamy. So one fertilization taking place here. Already there is a fertilization, there is a fusion, becomes polar nuclei and forms as a secondary nuclei. So one fusion already taking place. Now this secondary nuclei is 2N. So next fusion, next male gamete fuses with this secondary nucleus. So one of the male gamete, that is N, haploid, which fuses with the secondary nucleus, that is 2N. So N plus 2N, there is 3N. Now after this fusion, it forms the endosperm. Primary endosperm nucleus is formed. So endosperm is formed. So now this is 3n in number. Primary endosperm nucleus or the endosperm which forms the endosperm. The endosperm is 3n in number. Zygote is 2n, male gamete is n, and this secondary nucleus is 2n. One word question which is very important. And this zygote which develops into the embryo, and the endosperm also, it becomes embryo. From the embryo it produces the seed. So this is the process. There are two fertilization taking place. So the process or the phenomenon is known as double fertilization. And here if 3 nuclei are involved in fusion, because one fusion already in the polar nucleus, so this process is called as triple fusion. Triple fusion. So the name of the process is double fertilization and triple fusion. Double fertilization and fusion is 3 times triple fusion. Double fertilization and triple fusion. So this is the entire process when this process of fertilization observed in angiosperms. So this process, when this pollen germinates from this pollen grain towards this ovule, the germination of pollen tube, starting from here till the entry of pollen towards the micropylar region is called pollen pistil interaction. You know, this is the pistil which contains stigma, style and ovary. So pollen pistil interaction. This process is pollen pistil interaction. The germination of pollen tube from the stigmatic region up to this entry of ovule is called pollen pistil interaction. So this is the entire process, the fertilization, the event of fertilization. Just recall what are the events of fertilization. There is a first event is this deposition of pollen grain on the stigmatic region and germination of pollen grain, growth of the pollen grain and entry of pollen grain and discharge of the contents. That means the male gamete, tube nucleus, everything is discharged into the embryo sac, one of the synergies, that is egg. So this is the entire process, and this is observed in angiosperm, and the type of fertilization observed in angiosperm is double fertilization, and the process is triple fusion because of this 3 different fusions taking place. Hope you understand this very clear. Learn this process. It is the very important observed in angiosperms. Thank you.